In organic chemistry, we learned that carboxylic acid and amine could be combined to form an amide. So this is an amide. In the same way, the carboxylate group of one amino acid, for example, glycine, can combine with the amino group of a second amino acid molecule, like, for instance, alanine. This reaction takes place in the cell, and the product is a dipeptide. This dipeptide is called glycylalanine. This is the resulting peptide bond. It is important to realize that glycine and alanine could also be linked another way. In this case, we get a different dipeptide. Take note, it is the carboxyl group of alanine that combines with the amino group of glycine, and the resulting product is alanyl glycine. The earlier dipeptide, glycylalanine, is a constitutional isomer. Of course, they are different compounds in all respects with different properties. A dipeptide L-aspartyl-phenylalanine is of considerable commercial importance. The aspartyl residue has free, so this is the aspartyl residue, has a free amino group, the N-terminal end of the molecule. And the phenylalanine residue, ito yung phenylalanine, has the free carboxyl group. So this is the terminal end. This dipeptide is about 200 times sweeter than sugar. The methyl ester derivative of the earlier dipeptide has an ester linkage to the carboxyl group. Okay, so you call this an ester linkage. This resulting derivative is better known as aspartame or NutraSweet. This is marketed as sugar substitute under the trade name NutraSweet. The previous slide showed formation of a dipeptide. We can, of course, add a third amino acid to alanyl glycine. Like, for instance, we can add lysine. Then the product is a tripeptide. Because it also contains a carboxyl end and an amino end, we can continue the process to get a tetrapeptide, pentapeptide, etc., until we have a chain containing hundreds or even thousands of amino acids. These chains of amino acids are the proteins that serve so many important functions in living organisms. Okay, again, we identify in this tripeptide, there is the uh, amino terminus and the carboxyl terminus. So the C terminus or carboxy terminus contains the free carboxyl group, and the N terminus contains the free amino group. So this slide shows you the amino acids, the short peptides, the polypeptide, and the protein. The shortest chain are often simply called peptides. Longer ones are called polypeptides, and still longer ones are called protein. But chemists differ about where to draw the line. Many chemists use the terms polypeptide and protein almost interchangeably. In this presentation, we will consider a protein to be a polypeptide chain that contains a minimum of 30 to 50 amino acids. The amino acids in a chain are often called residues. It is customary to use either one letter or three-letter abbreviation shown in the earlier table, table 14.1, to represent peptides and proteins. For example, the tripeptide shown on the previous slides, alanine glycyl lysine, is represented as AGK, or this is the single letter abbreviation, A for alanine, G for glycine, and K for lysine, or the three letter abbreviation ALA gly ly. So we start naming from the amino acid that is in the N terminus going to the amino acid C terminus. The properties of proteins are based on properties of the peptide backbones and properties of the side chain. The peptide backbone consists of the repeating structure shown by the line of atoms in this figure. So this is the peptide backbone. Okay, it consists of repeating NCC, 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 and so on. By convention, peptides are shown with the N terminus on the left and the C terminus on the right. The 20 different amino acid side chains supply variety and determine the rest of the physical and chemical properties of proteins. Among these properties, acid-base behavior is one of the most important. Like amino acids, proteins behave as two-vetor ions. The side chains of glutamic and aspartic acids provide acidic groups, whereas lysine and arginine provide basic groups. The isoelectric point of a protein occurs at the pH at which there are an equal number of positive and negative charges. This is the point where the protein has no net charge. Here is a schematic diagram of a protein at its isoelectric point, at acidic condition, and at basic condition. The water solubility of large molecules such as proteins often depends on the repulsive forces between like charges on their surfaces. When protein molecules are at a pH at which they have a net positive or negative charge. Okay, so at isoelectric point, there is no net positive or negative charge. Ibig sabihin, equal ang positive and negative charges. At acidic condition, mag-prevail yung positive charge. At basic condition, mag-prevail yung negative charge. Okay, so when the protein has a net charge, kunyari, my positive or my negative charge, it becomes soluble. Okay, when the protein molecules are at a pH at which they have a net positive or negative charge, the presence of these like charges causes the protein molecules to repel one another. These repulsive forces are smallest at the isoelectric point, when the net charges are zero. When there are no repulsive forces, the protein molecules tend to clump together to form aggregates of two or more molecules, reducing their solubility. As a consequence, proteins are least soluble in water at their isoelectric point and can be precipitated. That is why in the laboratory, there is a technique called isoelectric precipitation of protein. The sequence of amino acid in the polypeptide chain is called the primary structure. The correct position of amino acids in the primary structure is important. Otherwise, it may result in a metabolic disorder like in the case of sickle cell anemia. So in this figure, we have here the sequence of amino acids in the normal red blood cells. It has glutamic acid in position number 6. In the case of sickle cell anemia, the glutamic acid is replaced by valine. So it results in a distorted red blood cell structure. The secondary structure of protein refers to the repetitive conformation of the protein backbone. Shown here are structures such as the alpha helix and the beta sheet. So yung repetition nito, yung paulit-ulit na pag-circle sa alpha helix. Dito naman is formation of a sheet. Shown here is the stabilizing interactions in the secondary structure of protein. Take note, sa alpha helix you have H bonds, H bonds. Likewise, sa beta sheet you have H bonds, and so on. The tertiary structure of protein is the three-dimensional arrangement of every atom in the molecule. Unlike the secondary structure, it includes interaction of the side chains and not just the peptide backbone. In general, tertiary structures are stabilized 5 ways. Some of the interactions that stabilize the tertiary structures. So we have here polar residues. So this is a dipole-dipole interaction. These residues are charged, my positive, my negative charge. So this is ionic interaction or salt bridge. This residue is hydrophobic. This is nonpolar, nonpolar. So this is hydrophobic interaction. And this one involves a metal ion coordination. Not shown here is the disulfide linkage, but it has been shown earlier. The quaternary structure refers to the spatial relationship or relationship in space, an interaction between subunits in a protein that has more than one polypeptide chain. Hemoglobin in adult humans is made of 4 chains called globins. there are two identical alpha chain composed of 141 amino acid residues and two identical beta chains composed of 146 amino acid residues. And this ends the presentation.